students, myself Bhupinder Kaur, lecturer Biology from Government Model Senior Secondary School 3B1 Mohali. Dear students, we are doing our 9th chapter, that is Biomolecules, and our today's topic is lipids. Students, we all know that biomolecules, they are of two types, organic and inorganic biomolecules. Organic biomolecules, they are made up of elements carbon, hydrogen and oxygen. So lipids, like carbohydrates, they are an example of organic biomolecules because they are made up of carbon, hydrogen and oxygen. Lipids, they are greasy and they are insoluble in water, which is a polar solvent, and they are soluble in non-polar solvents like ether, ester and acetone. Now, 1 gram of fats or lipids upon oxidation gives 9 calories of energy, whereas 1 gram of carbohydrates gives merely 4 calories of energy upon oxidation. Now, what is the reason why fats they give more energy as compared to carbohydrates? The reason is that the fats they contain less amount of oxygen in themselves. So they need more molecular oxygen for oxidation and therefore they liberate more energy. So students, let's start this interesting topic, lipids.
Dear students, the word lipids is derived from Greek word lipos, which means fats. So lipids, they are organic biomolecules, and these lipids, they are composed of long hydrocarbon chains. Lipids, they are made up of long hydrocarbon chains, that is CH2 chains. And these lipids, they hold an incredible amount of energy in themselves. Therefore, the lipids along with carbohydrates, they are known as energy giving foods. And they are an important component of our cell membrane. For example, cholesterol and other sterols, they play an important role, or they are necessary components of our cell membrane. Now these lipids, they are of three types: simple lipids, complex lipids, and third is the derived lipids. Simple lipids, they are further of two types: true fats and waxes. Complex lipids, they are of three types: phospholipids, glycolipids and lipoproteins. And derived lipids, they are of two types: steroids and terpenes. So let's discuss all of them one by one. First is simple lipids. Simple lipids, they are esters of fatty acids with alcohol. Now what is an ester? When an acid, it combines with an alcohol, then an ester is formed. So simple lipids, they are also esters of fatty acids with alcohol. These simple lipids, they are of two types: neutral or true fats, and second is waxes. Now what is the difference between true fats and waxes? True fats, they are esters of fatty acid with glycerol, whereas waxes, these are esters of fatty acid with alcohol other than glycerol. So if an ester is formed between fatty acid and glycerol, it is known as true fat, and if an ester is formed between fatty acid with alcohol other than glycerol, then it is known as wax. Now the true fats or neutral fats, they are made up of carbon, hydrogen and oxygen, and we all know that oxygen atoms, that is much less in fats as compared to carbohydrates. True fat is having two components. One is alcohol, that is glycerol, and the second is fatty acids. Now what are glycerols? Glycerols, they are actually alcohol, and they have three carbons, each bearing a hydroxyl or OH group. So this is the structure of glycerol with three hydroxyl group attached to three different carbon atoms. And the second component is fatty acid. Fatty acid is an unbranched chain of carbon atoms. and it has carboxyl group, that is COOH group at one end, and a hydrogen bonded to almost all the carbon atoms. So it is having carboxyl group, that is COOH group at one end, and hydrogen atoms, they are bonded to all the carbon atoms. Now these fatty acids, they are of two types. First is saturated and the second is unsaturated fatty acid. Now what are saturated fatty acids? In saturated fatty acids, all carbon atoms, they are linked by single bonds. There is no double bond. As there is no double bond, so they cannot combine with more hydrogen atoms. Therefore, they are known as saturated because they are already saturated with hydrogen atoms. Whereas in case of unsaturated fatty acids, double bonds, they are present between carbon atoms. So because of the presence of these double bonds, they can combine with more hydrogen atoms. Therefore, they are known as unsaturated fatty acids. So this is the structure of saturated fatty acid. A saturated fatty acid is having no double bonds in the CH chain, that is hydrocarbon chain, whereas the unsaturated fatty acids, they are having double bonds between the hydrocarbon chain or the carbon atoms. PUFA, that is polyunsaturated fatty acids, they are having more than one double bonds between the hydrocarbon chain. Now what are the various examples of saturated and unsaturated fatty acids? Lauric acid, palmitic acid and stearic acid. These are the examples of saturated fatty acids because there is no double bond between the carbon atoms in the hydrocarbon chain. Whereas oleic acid, linoleic acid and linolenic acid, they are the example of unsaturated fatty acids because they are having double bonds between the carbon atoms in the long hydrocarbon chain. Like we can see here that in oleic acid there is single double bond. In case of linoleic acid, there are two double bonds, whereas in case of linoleic acid, there are three double bonds between carbon atoms in the long hydrocarbon chain. So we can say that linoleic and linolenic, they are the example of PUFA, that is polyunsaturated fatty acids, because they are having more than one double bonds between the carbon chains. Now what are triglycerides? Triglycerides, they are a major class of lipids and it includes fats and oils. And the triglycerides, they are also known by the name fats and oils, and they are having the same components, that is fatty acids and glycerol. Now we will see that how these fatty acids, they combine with glycerol to form what? To form fats or oils, which are actually triglyceride. So this is the structure of glycerol, and this glycerol is having 3 hydroxyl group. Now these 3 hydroxyl group of glycerol, they will combine with the 3 fatty acids, and the OH group of the alcohol will combine with the COOH group, that is the carboxyl group of the fatty acids, and they will form esters. They will form esters, and these esters, they are actually triglycerides. So here we are seeing that 3 molecules of oleic acid, that is fatty acid, that is combining with 1 molecule of glycerol to form what? To form triolene, which is an example of triglyceride. So here there are various examples of fats. So one are pure fats and the second is mixed fats. Now what are pure fats? For example, triolene, this one, triolene triglyceride, this triolene triglyceride, that is having the same fatty acids, that is oleic acid. Triolene is formed from the same fatty acid, that is oleic acid. Therefore, triolene is an example of pure fat. Similarly, tripalmitin, it is having 3 molecules of only palmitic acid. Therefore, it is also an example of pure fat. But now look at the structure of palmitoyleosterine. This palmitoyleosterine, that is having 3 different fatty acids. One is palmitic acid, one is oleic acid, and one is stearic acid. So this is an example of mixed fat. Dear student, butter is an example of mixed fat because it is formed of 3 different types of fatty acids and glycerol. So a triglyceride is having 3 fatty acids plus 1 alcohol, and when 2 fatty acids they combine with 1 molecule of glycerol, it is known as diglyceride, and when 1 molecule of fatty acid it combines with 1 molecule of glycerol, it is known as monoglyceride. So these are the different forms of fats: triglycerides, diglycerides and monoglycerides. Monoglycerides, they are having only 1 fatty acids combined with glycerol. Diglyceride, they are having 2 molecules of fatty acids combined with glycerol, whereas triglyceride, they are having 3 molecules of fatty acids combined with 1 molecule of glycerol. Now, what are the differences between saturated and unsaturated fats? The saturated fats, they are made up of saturated fatty acids. So they have no double bonds between the carbon atoms of the fatty acid chain. Whereas the unsaturated fats, they are made up of unsaturated fatty acids. So they contain more than one double bonds between the fatty acid chains. The saturated fats, they have high melting point and they exist as solid at room temperature because they are made up of saturated fatty acids. Whereas the unsaturated fats, they are having lower melting points and they exist as liquid at room temperature. Saturated fats, they cannot be hydrogenated because they are already saturated, whereas unsaturated fats, they can be hydrogenated. Saturated fats, they have high cholesterol content, whereas unsaturated fats, they are having low cholesterol content. Now, what are essential fatty acids? Now we have studied in our previous lecture that there are essential and non-essential amino acids. Essential amino acids, they were those which our body cannot synthesize and for which we are dependent on diet. Similarly, there are essential fatty acids which our body cannot manufacture and for this we are dependent on diet. So all the unsaturated fatty acids like linoleic, linolenic and arachidoic acid, they are an example of essential fatty acids because these fatty acids, they are not formed within our body and we are dependent especially on non-vegetarian diets for fulfilling the essential fatty acids. Now second type of simple lipid is wax, and we all know that waxes, they are the esters of fatty acids with alcohol other than glycerol. Especially the alcohols which are involved in the formation of waxes, they are having high molecular weight. Now these waxes, they are of different types or there are various examples of these waxes. First is the beeswax. Beeswax is a secretion of abdominal glands of worker honeybees. Second, wool fat or lanolin. Lanolin is a secretion of cutaneous glands and it is obtained from the wool of sheep. Third is sebum. Sebum is secreted by the sebaceous glands of the skin, oil secreting glands of the skin. Cerumen is the earwax, which is present in our ears. Next is the plant wax. Plant wax is a coating formed on the plant organs to prevent the process of transpiration. And last is the paraffin wax, which is a translucent waxy substance obtained from petroleum. So petroleum jelly which we use in our home, that is an example of paraffin wax. So waxes, they form a coating over the surface of the leaves, and therefore the leaves, they are unwettable. They don't gain the weight when they are submerged in water. They are unwettable, and due to the presence of this waxy coating on the surface of leaf, the process of transpiration, that is lowered in plants. Next, complex lipids. Complex lipids, they are the derivatives of simple lipids having additional group, phosphate group, nitrogen base or protein molecules. And depending upon the type of the additional group, they are of different types: phospholipids, glycolipids and lipoproteins. So first are phospholipids. Phospholipids, they are made up of a molecule of glycerol or other alcohol having first a phosphate group at one of its OH groups, second two fatty acids at the other two OH groups, and the third a nitrogen base which is attached to the phosphate group. So we can say that phospholipids, they are having one glycerol molecule, two fatty acids, one phosphate group and one nitrogen containing base. And these phospholipids, they form our cell membrane, and they are having two parts. One is the long hydrophobic tail which is formed of fatty acid, and the second is hydrophilic head which is formed by the phosphate group. And we all know that these phospholipids, they are present in bilayer, and they are an important component of our cell membrane. Our cell membrane, that is formed of phospholipids. Next is the glycolipid. Glycolipid, they are the components of cell membranes, particularly the myelin sheath which covers the nerve fibers, that is made up of glycolipids. Similarly, the chloroplast membrane, that is also made up of glycolipids. Cerebrocyte is the most simplest form of glycolipids. So when the lipids, they combine with carbohydrates, they become glycolipids, and glycolipids, they are present in the myelin sheath which cover our nerve fibers. For example, we can see here that glucose molecule, that is associated with one end of the alcohol in the glycolipid cerebrocyte. So glucose or galactose molecules, they are associated with the glycolipid cerebrocyte. Next, third one is the lipoproteins. So lipoproteins, they are also an example of or a type of complex lipids, and they have proteins along with the lipid part, and they are main constituents of cell membranes, and they are found in milk and egg yolk. Lipids, they are transported in blood and lymph as lipoproteins. So whenever there is transportation of lipids within our body, they are transported in the form of lipoproteins. And these lipoproteins, they are of different types, like low density lipoproteins or high density lipoproteins. Low density lipoproteins, that is also known by the name bad cholesterol, whereas high density lipoproteins, they are known by the name good cholesterol. And the good cholesterol, it helps in the removal of bad cholesterol, that is LDL, from our body. Next, last is derived lipids. So derived lipids, they are the derivatives obtained on the hydrolysis of simple and complex lipids. So these are the derivatives of simple and complex lipids after their hydrolysis. And the most important example of derived lipid, that is the steroids. These steroids, they do not contain any fatty acids, but they are included in lipids because they have fat-like properties. And they are made up of four fused carbon rings, like we can see in this structure. All these steroids, they are made up of 1 2 3 4 fused carbon rings. For example, cholesterol, vitamin D, testosterone, which is a male sex hormone, and adrenocortical hormones, which are the secretions of adrenal glands. These are the examples of steroids. And the most common steroids, they are sterols. Common sterols, they are cholesterol and ergosterol. So all the steroids, they are having a common structure, that is the four fused carbon rings. Next, cholesterol. It is an example of sterol and it is a fat-like substance. It exists in animal foods and body cells. It does not exist in plant foods, and cholesterol is essential for the synthesis of bile salts, sex hormones, cortisone hormone and vitamin D. And it is needed by every cell in the body. Now, last form of derived lipids are the terpenes. Terpenes, they are major component of essential oils which are produced by the plants. And these essential oils, they give fragrance to the plant parts. Vitamin A, E and K, they contain a terpenoid which is known as phytol. Carotenoid pigment, that is a precursor of vitamin A. Carotenoid pigments or carotenes, they help in the formation of vitamin A and they are an example of terpenes. Similarly, a red color pigment lycopene, which is present in tomatoes, that is also an example of terpenoid. Gibberellins, which are plant growth hormones, they are also an example of terpenes. So next, in last, we have functions of lipids. What are the functions of lipids? First is the storage form of energy. We all know that lipids, they are energy giving foods, so they store energy within themselves, and because of their high energy value and uh, because many vitamins, they are soluble in fats, so they are very essential uh, components of our diet. Next, they are an important components of our cell membranes, like we have seen that phospholipids and cholesterol, they are an important component of cell membrane. They serve as thermal regulators or thermal insulators in the subcutaneous tissue एंड अराउंड सर्टेन ऑर्गन्स. सो ये इंसुलेटर्स का काम भी करते हैं, बॉडी हीट को लॉस करने से प्रिवेंट करते हैं. सो दे प्रिवेंट द लॉस ऑफ बॉडी हीट बाय एक्टिंग एज एन इंसुलेटर्स. नेक्स्ट, नॉन पोलर लिपिड्स, they act as electrical insulators, allowing rapid propagation of depolarization waves along myelinated nerves. So like we have seen that cerebrocyte, which is a glycoprotein, it is a covering around the nerve fibers, and these nerve fibers, they are known as myelinated nerves. So they help in rapid propagation of depolarization wave. Next, they provide shape and contour to the body. They act as metabolic regulators also. And these lipids, after combining with proteins, they form lipoproteins. And we all know that these lipoproteins, they are of two types: low density lipoproteins and high density lipoproteins. And they help in the uh, transportation of lipids in the body. So these lipids, they are transported in the blood only when these lipids, they combine with the proteins. So students, this was all about the topic lipids, and we have done in detail about the various types of lipids, what are their example, the structure of lipids, and now we are left with the function sources. What are the various sources of lipids? So first is plant sources, for example vegetable oil, margarine. These are the examples of uh, lipids which are obtained from plants. Nuts, olives, chocolate, avocados, these are all the sources of lipids from plants. Similarly, fatty meats, fish, dairy products, whole milk and egg yolk, they are the sources of lipids which are obtained from animals. So student, we have discussed in our today's topic in detail about lipids, what is the structure of lipids, what are the different types of lipids, and what are the various functions of lipids. So students, thank you.